Today, Sheila and I are going to present sweating the small stuff, so let's just jump right into it. So we have a couple discussion goals today. The first is to describe and provide instructions for you on the use of homemade as well as commercially available gels. And we're showing this to show you how we simplify the embedding of challenging very small tissues or cells. The procedures developed for handling these demanding tissues were done from specific cases that we encountered in our laboratory. And we're going to describe each one of these from the beginning stage, from wet tissue to histopathology analysis. So welcome. We don't need to go through introductions again, but when you hear this voice, this is Jennifer Johnson.
And this is Sheila Cummings. And so we'll get into it. Why is it important to evaluate disease in mouse models? So animals are used to assist in research and discovery as comparative models of human disease. And by far the most common animals used in research, as you can see in the pie chart in the upper right-hand corner, are mice and rats. And there are disadvantages and advantages to their use. So the disadvantages include the fact that they're live animals, and so their use must be respected and justified in every regard. In addition, their translational value or their similarity to the human counterpart of the disease may have important and inherent differences that need to be realized. But they have many advantages as well. They have very similar homology with the human genome, and strains of mice are genetically homogeneous, unlike the human population. They're small and less expensive than any other research models like dogs or non-human primates in particular. They can be genetically engineered to be very similar to some of the diseases that we'll discuss today. And so why are we talking about this today?
Well, it all comes down to evolution of an idea. For those of you who are in research, and of course this also applies to those of you doing clinical work, your work requires creativity. And so one of my responsibilities is to improve the histology by working out better ways to use our different processes. And I do this in order to more specifically evaluate the models that were given and to achieve specific research goals. So, like we said, we're going to go through this from the point where we received our quote-unquote missions all the way to the end when we have glass slides. So we're going to go back a few years, and upon my arrival at work that day, I was informed that I'll be providing histology support in the characterization of a mouse model, and that's going to be used for testing therapeutics for Niemann-Pick disease. The mouse model is a knockout mouse. That means they created the mouse model by knocking out the same gene in the mouse that's been shown to be defective in the humans who have the disease. So as usual, I start researching this rare disease and find there's only a little information out there published describing the human disease, and I can't find anything except one publication on the original mouse model that I'm about to start working with.
But first, what do you know about Niemann-Pick disease in humans? Niemann-Pick disease is what's known as a lysosomal storage disease. Lysosomal storage diseases are a group of more than 40 diseases. Each of these is caused by some sort of genetic problem resulting from the deficiency or malfunction of a particular enzyme, which is needed to remove waste material from cells. As the waste molecules accumulate in the cell's lysosomes, which are little organelles within particular cells, the cellular function can be disrupted. And this can result in a variety of symptoms depending on which cell type is affected and what tissue is affected. Eventually, it will affect the entire organ. All LSDs are progressive, and that means that without treatment, they will worsen over time. Niemann-Pick is an inherited lysosomal storage disease, and it's due to the genetic mutation of these particular genes. These mutations result in an enzyme deficiency of what's known as ASM, or acid sphingomyelinase. It's normally used by cells to break down lipids. So without the ASM, the lipid can't be broken down, and it accumulates in those organelles called lysosomes in the cells. The accumulation eventually affects the tissue and organ function, and all forms of Niemann-Pick disease are progressive, leading to worsening symptoms and poor prognosis. There is a mouse model for Niemann-Pick disease, which, as Jen mentioned, is a knockout. It was designed, and this is the paper where it's referenced the genetic makeup of this mouse. It's deficient in the same enzyme that is missing in the humans, and it recapitulates the sphingomyelin accumulation very similarly to what's seen in the humans. The tissues that will be affected will include the brain, the spleen, the liver, and the lung. And the lung is the tissue that we'll be investigating today.
So the goal was to determine if there's disease comparable to the humans in the lungs of the mouse. So in addition to taking sections of the lungs, I've been asked to obtain lavage cells from the mice to be examined by H&E and IHC as well for the pathologist to determine the cell type. And this is going to be important because we want to be able to compare the lavages from these human patients to assess the disease and evaluate the effect of any future treatments. So my job was to obtain the cells from the lungs of these mice and generate the slides to allow all of the above to happen. So as usual, I have to come up with a plan. So here it is. At necropsy, I was going to lavage the lungs with fixative and collect the cells. The cells would then be embedded in agarose. The agarose blocks can then be processed into paraffin, and this will allow us to take multiple sections that can be stained with everything that the pathologist is requesting, leading to a happy pathologist.
So a bronchoalveolar lavage is a bronchial washing using saline solution, usually in humans and animal patients that are alive. And it's a non-invasive way to collect cell types that are deep in the lung and indicative of certain diseases. They're used in the diagnosis of lung cancer, pneumonia, parasites, opportunistic infections, and inflammatory airway diseases. So just as an example, the center image at the bottom of the slide shows a horse with a COPD, and we look for particular mast cells and particular cells that would help us diagnose COPD. And then on the right are lungworms that were obtained through bronchoalveolar lavage from a cat. So just to give you an example of veterinary application.
So from lung to test tube, the mice were lavaged and the cells were collected in fixative, according to plan. I allowed the cells to fix undisturbed for 24 hours in the tube, and I used neutral buffered formalin as my fixative here. And if the cells are not already in a pellet form after 24 hours, or if they appear to be a little more flocculent than solid, that is kind of floating within the liquid, I would spin them down at room temperature for about 5 to 10 minutes at 1,000 to 2,000 RPMs. So we have to make our homemade gel, which is a 2% agarose. Agarose is a polysaccharide that's extracted from seaweed. It's available as a white powder, which dissolves in near boiling water, and it forms a gel when it cools. Our lab currently uses it at a 2% concentration, and it's made in water, to embed cells. If we're going to use it for electron microscopy, we use a 2.5% concentration, but we're not going to get into that today. It is prepared in advance, and I store it in a covered sealed test tube in the refrigerator, and I warm it up just before use. So let's go through how I prepare it. I'll weigh out 2 grams of agarose per 100 mLs of filtered water, and then I'll warm it in a glass flask on a magnetic stir plate with stirring, and I always make sure I use a much bigger flask than the volume I'm making. I don't want to have any boilovers and messes, because nothing smells as bad as agarose when it's burning. So with the stir bar in motion, I will slowly add the agarose powder. If you add it all at once or before the water has started to warm, you end up getting clumps that take a long time to dissolve. So this is my little setup on the stir plate, and you can see I'm adding it slowly and stirring. Just imagine it stirring. So continuing, I'll keep mixing it until it begins to boil rapidly. If you look on the left, you can see it's opaque here. That's because it hasn't dissolved. But on the right, you see bubbles forming here. That's when it's starting to boil, and it turns clear. So that's when I'll turn down the temperature enough so it continues to boil, but just barely, and I let it cook for another 2 or 3 minutes longer, not too long. I'll turn off the heat, but I leave the stirring on. I want to let it cool enough that I can handle the flask and aliquot it out into smaller volumes. Those smaller volumes will get stored in sterile sealed tubes. I usually do 10 or 15 ml conical tubes. sometimes 50s, and I'll store them in the fridge for a month or two. So if you look down at the picture here on the left, you can see it's clear. That's a heated tube. On the right, you can see that solid opaque color. That's when the gel just comes out of the refrigerator. And so I usually check it for contamination when it is in that solid form because it's easier to see if there's any contamination there. And I'm very careful that when I'm performing the procedure itself, I only warm up what I need and throw out any warmed and unused material when I'm finished. Remember, when in doubt, throw it out. Once it's opened and warmed, it may have become contaminated, so it's better to throw it out than to have to go all the way back to the lab and start over collecting more tissues. So preparing the agarose for use and the actual embedding. So the day that I'm going to do it, I'm going to need to warm up my agarose prior to use. And I need to get it to about 60 degrees. I can tell it's at the right temperature when it turns clear again. That's how I know it's ready for use. There's a couple different ways to do it. Sometimes I'll put it in a beaker of water, warming it on a hot plate. And what I'll do is pick it out and kind of swirl it around a little to test for complete melting. You can also put the tube in a 60 to 65 degree water bath for about 30 to 60 minutes prior to its use. You can also microwave it on a low setting for 5 to 15 seconds at a time. It's really important to loosen that cap before heating it, because if you don't, the cap will pop off. Kind of sounds like popcorn if you have multiple tubes in there, and you may scare your lab mates. So I only do it 5 to 10 seconds at a time, and you need to check if it's melted in between. And you just repeat it until it's uniformly melted. One of the most important things, though, is to make sure it isn't too hot before you use it. So if it's too hot to hold that tube, then it's too hot to use. You just don't want to fry those cells. But it's important to keep it in the warm water. So I usually have a warm water bath nearby when I'm using it because it hardens pretty quickly. This is my little bath that I set up. I have the heat on low, and you can see there the one on the left is ready. The one on the right needs a little more time. So now I'm ready to embed. I'm going to remove the fixative with a pipette, and I'm going to be really careful not to disturb the pellet. So what I use for that is a plastic pasture pipette that I've added a pipette tip to. So this is a plastic 200 ml pipette tip, and I just put it on the top, and you can see it changes the blunt end of this to a nice pointed end, and I'm able to get down closer to the cells and to get all the liquid out. And to ensure that small pellets are not disturbed, it's important to use something like this, or even a Kim wipe. I kind of twist the Kim wipe and make a long, like a little stick with it, and I'll go in there to absorb the rest of the liquid, usually fixative, away from there. It is critical that you get out as much liquid as possible prior to adding that agarose. So I'm going to add my warmed agarose 1 drop at a time to the cell pellet. I'm going to use enough to completely surround the pellet, and then I'm going to add a few extra drops on top. If you can kind of make it out here, what I'm doing is I've got the pipette tip against the edge of the tube in order to slowly release these drops so I don't disturb my pellet. However, if your sample, as soon as you start adding your agarose, becomes flocculent, you only want to add a few drops and then stir to mix those cells into the agarose, so they're dispersed throughout the little drop of agarose there. Then you're going to cool it and allow it to harden completely. And the important thing here is to be patient. You can either do it on ice or in a refrigerator or just at room temperature, but I usually wait about 5 minutes when it's on ice, longer if it's going to be at room temperature. Then to remove them from the tube here, what I'm using is a waste spoon here. You can also use a wooden stick or something else. And basically what I'm doing is sort of getting it underneath and popping the gel up and out. There are times that I will also use an Eppendorf tube if I have a very small volume of cells. I'm going to pop it out, and then I'm going to put it into a cassette for processing. If it doesn't come out easy, then you need to put it back onto the cold, because it could be that it's just not solidified completely. It's just not cold enough. Or if that doesn't work, briefly dip the whole tube, make sure you put a cover back on top, into hot water and try again. And then if you've melted a little bit on the outside, sometimes it's a little easier to pop it out. Then, like I said, I'm going to place it into a cassette and get it ready to process. In our lab, gels are stored for only a short time, and usually they're in formalin in the refrigerator until it's processed. A couple do-nots here, because there's always a few do-nots, right? It's important not to leave it in PBS or saline because that gel will swell. It's also important not to leave it anywhere near anything warm. We found out that the hard way. It's also important not to leave it for a long time in anything, any solution. So for best results, you should try to process them as soon as possible. So usually I will try to schedule with the processors to do them that evening. A little troubleshooting, because it's always the small pieces that make up that big picture. If your samples break into smaller pieces while you're removing them, or the pieces are not big enough to process, you can use a biopsy cassette if you need to. If the pieces are too small or broken up to process comfortably, it could be due to the number of cells within the pellet. Next time you should probably add a little more gel. What you can do, though, is re-embed them. Dump those little pieces out onto a cold plate or a piece of dental wax, and then trim off any of the excess gel you can see that doesn't have any cells in it. And then re-embed them in fresh, warm agarose. If I have to do this, I usually use a regular embedding mold to do this. If the cells didn't stick or get encased by the gel, you can remove the gel, cut out any cells you see, and add the cells that were encased in the gel, as well as a little bit more agarose back to your tube and mix it to get the cells into the gel. And then you would process any and all pieces that get created. Well, what do you do if the gel doesn't form a solid pellet? That is, it looks more like jello or jelly than jello. Well, that means it's not completely cooled, possibly. You could throw it back in the fridge and see if that helps. If it's not, it may be that there was too much liquid that was left behind with the cells when the gel was added. So what you can do is gently warm the sample in a water bath until it's liquid. At this point, you really don't want to use the microwave because you don't want to fry your cells. Once they're warmed, you can spin the sample down in the gel at about 1,000 RPMs for 5 minutes. And if you have a heated centrifuge, that's the best way to do it. Remove the liquid and try embedding it again in fresh agarose and put the sample into the fridge or on ice to cool. So processing. What we're going to do is talk about processing at the end. So once we have all of these quote-unquote studies ready to process. So let's look at the next assignment. So while characterizing a mouse model for Hurler disease, you and your pathologist find that there's evidence of heart disease in the mice from this study.
But what do you know about Hurler syndrome in humans? Hurler syndrome is another inherited lysosomal storage disease. It results from a mutation in the IDUA gene, which causes a deficiency in an enzyme called alpha-L-iduronidase. So this enzyme would ordinarily break down mucopolysaccharides in lysosomes within certain cells. So without it, there's an accumulation of what's known as glycosaminoglycans, or GAGs, in the lysosomes within certain cells in the body. With the accumulation of GAGs, there is an interference with the normal functioning of cells, and it causes, again, a progressive damage to many tissues, including skeletal abnormalities, hepatosplenomegaly, which means enlarged livers and spleens, large hearts, or cardiomegaly, and valvular disease, among others. There is a mouse model for Hurler disease, which was, it's a knock-in, meaning the gene was knocked in to these mice generated at the University of Alabama. The mouse carries the mutation similar to that seen in the humans with the Hurler syndrome, and it really closely recapitulates the GAG accumulation and the multisystemic pathology that is seen in humans with Hurler syndrome. There is cardiac involvement in Hurler syndrome in humans, and it can be a common and a very early feature of the disease. So changes in the heart of afflicted patients can include abnormalities in the cardiac valves, the coronary arteries, among other pathologies. So we needed to investigate the features of the mouse model as it compared to the cardiac findings that were seen in humans with this condition.
So your challenge is mouse aortas. And what we needed to know is, first, is there disease present? So we needed to create H&Es and then to create slides that we could do special stains on to demonstrate the disease. And if so, is the disease focal or spread across the whole length of the aorta? The pathologist will need to evaluate multiple sections through the aorta for every animal. But before we get into that, I'd like to introduce you to Dinesh. This is one of our other pathologists here, and he is famous for leaving these wonderful little drawings on my desk. And I have to somehow bring his drawings to life. And this is what was on my desk, on a paper towel, the word aorta, and then some little rectangles and arrows. But I got it. I knew what he was trying to figure out here. But how was I going to get multiple sections of these thin pieces of this thoracic aorta into a section? I didn't want them to collapse or get damaged during the processing or embedding. How was I going to do that? Well, as usual, got to start with a plan. My plan was to euthanize the animals and collect the aortas in formalin to be able to embed them in paraffin, and that way I could get sections to perform both H&Es and special stains. But instead of trimming them into little pieces at the embedding station, which is what we would usually do, I decided to embed them into histogel. But if you can see here, this is a mouse aorta right here, and it's about 1 millimeter wide, and it's probably about 2 centimeters long here. So you can imagine that this is a very fragile and delicate tissue, and handling it really can create artifacts that will make analysis difficult for the pathologist. So I decided to use histogel. And what is histogel? Well, the manufacturer calls it an aqueous gel composition useful in processing histological and cytological specimens. That's a mouthful, but really it's just commercially made histology jello. It's a solid at room temperature, and it has that nice pink tint to it before processing. After processing, it comes out clear. But like agarose, it must be liquefied for use, and the manufacturer says to heat it to 60 to plus minus 5 degrees. So the manufacturer directs warming it by microwave, and similarly on a low setting for 5 to 15 seconds at a time, and checking in between. And again, loosen that cap. Or to put it into a boiling water bath for 3 to 10 minutes. The manufacturer also says it will stay liquid at about 50 degrees. And I say, remember, if the tube is too hot to hold, it's too hot to use. So this is my embedding station. I have a small styrofoam box that I got some antibodies in. And what I do is I fill it with ice, and I have an aluminum block that I will keep in the freezer. And I usually use clean metal embedding molds here to do the embedding of the aortas. So the embedding of these thoracic aortas to get those multiple cross sections actually is a two-step process. So first, I'm going to add enough warmed histogel to cover the bottom of the mold completely at a 1 to 2 millimeter depth. And then I'm going to allow about 10 seconds for it to harden. Then I'm going to remove the tissue from fixative and blot it on a paper or Kim wipe because I want to remove as much extra liquid around it as possible. Then I'm going to orient it on the histogel and cover it with a layer of warmed histogel. And then I'm going to allow the block to harden completely before touching it. That is, by keeping it on that aluminum block. You could also use ice or just do it at room temperature. The manufacturer says to leave it for 2 to 3 minutes, but it doesn't specify what volume or what temperature. I usually try to wait at least 5 to 10 minutes. And this is what it looks like when the first embedding is complete. You can see there's a little space between the reflection here because there's a layer of histogel at the bottom there. And that's important because I really want to encase the whole aorta in the gel. So trimming the embedded aorta, the next thing I need to do is pop the whole thing out. And you can see I'm using a little weigh spoon here to pop it out. And then I'm going to trim it using cold, sharp blades. So these razor blades I leave sitting in my ice, and I use a quick, decisive motion to cut the block and create several pieces. Before we go on to the second embedding, I just want to talk a little troubleshooting. So when you cut those aortas with the razor blades, if they come out of the histogel block, you can do a couple things. It's important, though, like I said before, to make sure that you blot that specimen completely because chances are it came out because there was a little bit of liquid left around it. So you can either carefully slide it back into the hole or you can put it into a new block of histogel. If the layers separate when cut, that is, You can see here there's sort of a separation here, and when I trimmed the block, these two pieces fell apart. Well, this was the bottom and this was the top that I poured over. So what happened is I actually waited a little too long after the first layer was put down to add the second layer. So what I'm going to do is remove the tissue and I'm going to try embedding them again. Moving on to the second embedding, I'm going to add a little warmed histogel to the bottom of a clean mold and put the gel blocks in one at a time, face down. You can see it creates a cross section. You can see the lumen there, that little hole in the center of the aorta there. Then I'm going to add more warm histogel around the blocks, and that basically glues my little blocks together. Then I'm going to cool and pop the block out and trim them so that they can fit into a cassette for me to process. And here's how I trim them, and usually I'll also add a notch to the top here, and that signifies that that is more toward the rostral end or the nose of the mouse, and the tail end is down here. And this is what it looks like in the cassette. So there are other ways to use this technique. We didn't end up just using it on the aortas. We also used it on the mouse spinal cords. So we had an investigator who wanted to look at multiple sections, that is 15 sections, of each of 3 different regions of the spinal cord to determine if lesions were present. Our original method was to take each of the 3 regions, so cervical, thoracic, and lumbar, and then process each one individually, embed 3 sections of each, form a block, and then cut 15 sections. Well, rather than cutting 3 blocks 15 times, what we did was embed all 3, cervical, thoracic, and lumbar, into the histogel, and then do our trimming similarly, our re-embedding of these, trimming again so I could tell, okay, I know my orientation here. And then we formed a block so that rather than cutting 3 blocks 15 times, we could cut 1 block at 5 different levels. Saved a lot of time. So still, along the lines of working with this hurler disease, I got another little piece of artwork from Dinesh. I think he's actually pretty good here, but you can see here's the mouse heart, and here is the aorta here and the descending aorta. And what we were finding were these lesions were present in the aortic arch of these animals. And so Dinesh wanted to look at this aortic arch as well as where it came into the mouse's heart. So a little warning here, this is actually a gross photo, and it's kind of gross. So as I said, the aortic arch is affected. So here is the heart of the mouse, here's lung here, and here is the aortic arch. And you can see it's red here, filled with blood, and this is distended. It should not look like this. It should be a small, clean, pink tube. What you're seeing over here is a clot of blood, and what has happened is that due to the instability of this damaged aorta, the aorta has ruptured, and this animal died due to the bleed here. So in the past, it was difficult to get that correct orientation that Dinesh wanted for analysis because the hearts would warp during processing. Anyone who's worked with mouse tissues knows. Mouse tissues are not just dry, but they also tend to warp a little bit. After processing, that aortic arch was no longer in line with the cut face of the heart as we needed it. And if you press it to flatten it, that brittle little heart tissue would break. So how can you ensure that correct orientation of the tissues and protect them from getting broken during processing and embedding? Well, we decided to try embedding them in gels. So prepping before gelling. This is one line here, dissect out the hearts and aortas, but I'll tell you that one line took hours. They were then trimmed to determine that correct orientation that we talked about, and this is a picture of that orientation. So here are the ventricles down here in the lower part of the heart and the atria up here, and attached here is that aortic arch that we wanted to look at, along with the descending aorta. You'll notice that there were holes in that tissue, the atria are holes, the ventricles, the aorta. Those need to be filled with the gel because air bubbles in paraffin are bad news. We've all tried to cut around those, right? It's no fun. So in order to start the embedding process, I have to prep and warm my gel and then get out the embedding molds and set up that same chilling setup that I showed you before. Getting them into gel involved quite a few steps. First, I'd blot off as much fixative from the specimen as possible, and then I would drop my specimen into some warmed histogel and just kind of mix it around a little bit. Then I would go to my embedding mold and add enough warmed histogel to cover the bottom of the mold, again at about a 1 to 2 millimeter depth. And then I'd quickly pluck out my heart and aorta from that warm histogel and orient it on the tissue, on the gel in the mold. And then I'm going to cover it with a layer of warm gel. I'm going to make sure that there are no bubbles in there. And then I'm going to allow that block to harden completely before trying to touch it. So again, keeping the block on ice or at room temperature, either way, be patient and wait the 5 to 10 minutes. Then I'm going to trim away the excess gel, cassette, and process it. And this is what they look like. They're actually pretty cool looking. Yeah, I kind of like that. And you'll notice I used a clear embedding mold. This is one of the cryo molds that we have in our lab. And I did that because I want to be able to see and make sure that there are no bubbles in these holes here. So now on to the last challenge. We'll get to processing all those other ones together in just a few minutes. So there was a very large study that we were investigating Fabry disease on, and this study was to evaluate skin samples which have had a history of problems. But first,
so what do you know about Fabry disease in humans? Again, Fabry disease is a rare inherited LSD. It's due to a deficiency in the enzyme alpha-galactosidase, and this disease is X-linked, meaning that it primarily affects males. So without the alpha-galactosidase, there's a fatty substance called globotriacylceramide that cannot be degraded, and it tends to accumulate, again, in the lysosomes of cells. So the accumulation of GL3 within the lysosomes tends, again, similar to the other lysosomal storage diseases that we discussed, it causes cells to swell, eventually affecting tissue and organ function. Fabry disease generally affects the heart, kidneys, brain, and GI systems, as well as the skin of patients. These patients can suffer from angiokeratomas, hypohidrosis, which is like an inability to sweat, and painful acroparesthesia, which is kind of a painful pins and needles in their skin due to the accumulation of lipid that occurs in vessels and the nerves of their skin. So there is a mouse model for Fabry disease. It's a knockout mouse missing the gene for alpha-galactosidase. There's a progressive accumulation of GL3 in multiple tissues of this mouse model, which recapitulates the accumulation and multisystemic pathology that's seen in humans. Mice, however, do not have clinical disease as you know, the human patients do, and they do have a normal lifespan. So the disease is less severe in the mouse model than in human patients. But we wanted to investigate the foot pads in our current study, and it was a fairly large study, as there seems to be an accumulation of GL3 in the foot. So in previous studies, they weren't able to get a good look at the skin samples of the feet taken from different regions, back and foot pads, due to technical issues. Like, as an example, the image down at the center of the slide shows ears, mouse ears. So the center has a band of cartilage, and because the tissues are so different, you know, you have cartilage, you have skeletal muscle, you have adipose, you have epidermis. All of these different textures of the tissue tend to create artifacts, and sometimes those artifacts are really hard to read around as a pathologist. So the goal here was to avoid some of these curling and fixation and processing artifacts and try to get some cross sections of the feet and back tissues and avoid some of that separation.
So your last challenge is these mouse skin samples. How can you assure perfect cross-section orientation and still have the embedders like you? Like Sheila said, this was a large study. I think there were 60 animals. We were taking 2 samples from the back and 2 samples from the feet. And so the embedders had to stand all of these little pieces exactly straight up and down as best possible. So how can you improve the processing so that the samples don't separate on the water baths? Well, we use tissue orientation gels in order to do this. So again, got to come up with a plan. So I collected tissues and fixed them as straight and flat as possible. Now that was one of the issues that we had. And you can see here I have placed them onto paper towels. These are the paper towels that are everywhere. They're probably in your lab too, the trifold paper towels. And what I found is that the tissues are just a little wet and the paper towels very dry, and they tend to stick to them, so it was easier to put these tissues on here, and they remained straight during fixation. This made it easier for us down the road with embedding and processing and so on. So my plan was at necropsy to collect the foot pads and back skin as well, but we're just going to show you foot pads in this case. And then we're going to spread that tissue onto the paper towels to hold it flat during the fixation prior to cassetting. After they are processed, the foot pads are going to be trimmed to have a flat edge along one long face, and then we're going to slide these thin strips into the lanes of the tissue orientation gels. The gels are pushed close as much as possible. That would lead to successful orientation of the tissue. This was going to lead to happy embedders who only had to deal with one orientation gel per cassette, happy cutters because hopefully the processing would be improved, and a happy pathologist. And that's important, right, Sheila?
Yay!
So tissue orientation gels. I got these from Sakura. They're called paraffin gels, and the ones we decided to use were these 2-lane gels. And this is what the package looks like. So what I did is take the orientation gel, and I would spread them just a little bit with my forceps, and then I would slide in the mouse feet, the foot pads here, and then I would sort of push it closed a little bit, and then cassette them in either the regular cassettes or use the paraffin cassettes here. Those help to keep them even straighter, we found. So before we go into processing, I want to talk about why use agarose versus histo gel versus tissue orientation gels, and there's so many reasons. The first is cost. Obviously, agarose is the least expensive route to go here, but you have other factors like time. A tissue orientation gel is ready to go when your biopsy arrives or when your tissues are here. It takes a lot less time to work with, and so that, in a sense, saves you money. Reproducibility. It's important that no matter what you do, you're able to do it every single time. So whichever option you see there works best for you. There's also GLPs, SOPs, and documentation. Ugh, my favorite. It was a lot easier for us to use the histogel or the tissue orientation gels because we didn't have to track solutions and lot numbers and so on all the way back with the agarose preparation versus, you know, an already commercially prepared product. And there's also personal preference. You might like the texture, color, or the way something cuts or stains or works better. So individual preference is another reason. So let's talk about processing now. Once they're all in the gels, it's time to process them. And it seems pretty simple, right? But it is a little deceiving, we found out. So a little background before we get into processing. Our lab processes mostly mouse and rat tissues. There's some human, primate, and a smattering of rabbits and guinea pigs and so on as well. But as such, we usually have the luxury of time. That is, the fixation is done off of the processor prior to receipt. Our processors are set up with alcohols and xylenes and a low melting temperature paraffin. And by far, the small cycle is most often. The gel blocks looked small, so we processed them under the small cycle. And this is how they came out. That's not how it went in. You remember the gel was clear and kind of looked nice. It certainly didn't go in looking like this. Our small cycle is about 30 to 45 minutes per bath. And these examples here are actually fetal mouse femurs here. But why do they look like potato chips? It's all about the processing. So let's take a quick look at the processing. Everybody knows what fixation does. And you know that dehydration in alcohols is controlled by using this timed series of graded alcohols. And the goal is to remove that water, that excess water. And then you transition in clear and xylene and infiltrate with paraffin. But what if there's water left in your sample? So the water, as you know, if it comes out of your alcohol, isn't going to get absorbed by your xylenes. you end up seeing it at the bottom of your container or retort that you're processing. And this is what I found. So after processing, when I opened up the processor, this is what I found, all this condensation, and it didn't smell like xylene, and it didn't smell like alcohol. It was just water, because the water was remaining after clearing and going into the paraffin. As that paraffin is heated, the water kind of boils off. And I was boiling my tissues, and thus the potato chip effect. So if you're going to process your gels, we recommend using a medium or a large cycle, that is at least an hour to an hour and a half, depending on the size of your gels, in order to make sure you've got all that water out and so that they will process correctly. Embedding. After processing, the gels have to be pushed down as flat as you can into the bottom of the embedding molds. And you have to be careful of extra bubbles and spaces. Use a disposable pipette to force the paraffin into any holes or suck any air bubbles out. And don't let that gel cool prior to embedding it, or you'll get more bubbles, and bubbles lead to troubles. There are many advantages to using these gels at embedding. When you're embedding in gels, there's no chance of losing small tissues. When you open your cassettes or biopsy papers, have you ever done this? You have a small sample, you open it up, and the cassette cover comes off, and it goes flying across your lap, and you're looking around trying to find it. There's no way that that's going to happen. It's in a gel, and it's already, and it's in a gel that's large enough. There's no question about orientation either. These samples just get embedded flat. There's also no need to overhandle fine specimens that are prone to mechanical damage, like those spinal cords or thin nerves or those aortas. You only handle the gel itself. And the gels can prevent the curling and warping of samples like those aortas and hearts during the processing. This is going to lead to better sections. Multiple specimens can be put into one gel before processing so that only one object is manipulated at embedding. And all of these factors lead to speeding up the process at the embedding station, and happy embedders. So I just want to quickly show you what they look like at embedding. Here is the bottom of a conical tube, and you can kind of see the cells in here after processing. This is what they look like when we go to embed them. And here's a block, and you can faintly see and make out the bottom, the triangle here, but also faintly see the cells. And this is what one of the heart samples with aorta look like, and they're really kind of cool. This is what it looks like as it comes out of the processor, and here's an example of a block. And you can see we were able to get the gel inside here, and that's going to prevent bubbles and some of the sectioning problems. This is a set of lined up aortas, and I usually actually put them in at an angle just because of the size of the block. And last here we have what the tissue orientation gels look like, and we have the mouse foot pads sitting inside, and I usually do those at an angle as well. This is what they look like as a block. And as for sectioning, there's really nothing different than with regular blocks. The blocks have to be cold before cutting them. I like to use the slushy ice bath, but it's important not to let them soak too long. The thickness is really protocol dependent, what you're going to do with them after and how you're going to stain them. Ours are regularly cut at 5 microns. And then after they're taken out of the water bath, I usually let them drip and drain for 10 to 15 minutes and then bake at 60 degrees for about a half hour before staining. And here they are on the water bath. You can see they're still opaque here. I know it's kind of a crummy section there, but I just wanted to show you what they look like. This is what the agarose looks like. And as for staining in general, we use those same protocols we use to stain our tissues. There can be sometimes a little faint background of hematoxylin staining visible, but for our needs, it certainly has a hampered interpretation. So let's go through some of the results.
Okay, so as a recap, we'll discuss the Niemann-Pick disease lung lavage, Hurler syndrome aortas, Hurler syndrome aortic arches, and then the Fabry disease mouse skin from the foot pads. But first, I want to give a little anatomy lesson, okay? So here's a normal mouse lung under the microscope, and you can see there's a lot of air spaces here, and this is where, of course, oxygen exchange occurs. This is a big blood vessel right here, and here is probably one of the big bronchus, bronchioles from the mouse. But this is what you'd expect to see. And this is on a little higher magnification, so you have these tiny little alveolar septa. They're so small that you really have only one red blood cell fitting through each one of these little blood vessels here. And this is where, of course, your oxygen exchange occurs in that space. So this is a Niemann-Pick mouse lung, and you can see you might be able to appreciate it's a little busier than the other tissue that we were looking at. So that's because within some of those alveolar spaces, which should be clear like this, are filled with these very large foamy eosinophilic cells. And so that's what they look like side by side. So you have your Niemann-Pick mouse with those cells on the left and a normal on the right. So the bronchoalveolar lavage was to recover some of those cells so we could identify those. And so this is what they look like in our BAL and stained with H&E. And again, those are those big foamy cells. And what we did was use immunohistochemistry, an F480 stain, to identify these cells as macrophages. So the lysosomes were present in these macrophages, and that was causing the pathology in the lungs of those Niemann-Pick mice. So is the disease observed in the mouse model similar to that seen in the affected human lung tissue? Yes, it was. And did the lavage cells stained with H&E and F480 IHC help with this assessment? Yes, it did. So both of those missions were accomplished in this case. So in discussing the Hurler disease mouse aortas, on the left you have a Hurler mouse aorta cross-section, and on the right is a normal cross-section. And you might look at this area here on the lower left side of the Hurler mouse, and there's really some disorganization going on here, which we will identify in just a second. So the aorta really requires a lot of elasticity. With every contraction of the ventricles, a lot of blood is being ejected into this aorta, and it really needs to swell and then shrink again to accommodate the ejection of that blood. And along the wall of the aorta are these, you'll see these bright pink lines, and this is actually elastin, and this contributes to the ability of the aorta to stretch and then retract again. So on the right you have a normal mouse, and on the left we have this Hurler mouse. So in this case, we used an elastin stain to help identify the differences. And on the right, if you could see that all these elastin stained fibers in the wall of the mouse are continuous and regular. But in the Hurler mouse aorta, you can see that actually some of them are missing altogether, and they're thinner and irregular. And so that, of course, would contribute to what you saw in that gross mouse photo that Jen showed of the aortic rupture, and that aorta would be very friable and possibly rupture. And so this is one of the things we were able to identify with these beautiful sections. We also wanted to look at the aortic arches for a similar reason, because that pathology was identified not only in the aorta, but all along that aortic arch in particular. On the right is a normal mouse, and this is actually the aortic valve cross-section here, which is kind of cool. And then on the left is Hurler mouse. You can see that the thickness of the wall is very different and much more cellular. So is there evidence of histopathologic changes present in the aortas and the aortic arches of the mouse model? Yes, indeed. And if so, are the changes focal or diffusely present in the aorta? And we found that they were diffusely present in the aorta. So again, the mission was accomplished in these cases. So finally, we have the Fabry disease mouse foot pad. So this is the cross-section that we were showing you in the tissue orientation gel. And as Jen mentioned, there's like a very faint haze of the tissue orientation gel here, a pink from the hematoxylin. So you've got, but obviously that does not interfere with my reading of this tissue, and you can see that there's no separation between the epidermis and the other tissue components, the skeletal muscle, the adipose tissue. It's all nice and very even, and so that's it's a pleasure to read that tissue as opposed to an artifact-filled tissue. So were we able to get a perfect cross-section orientation and still have the embedders like us? Yes. And were we able to improve the processing so that the samples didn't separate on the water bath? Yes, and again, mission accomplished. So in conclusion, we described and provided instructions in the use of homemade and commercial gels, hopefully to simplify embedding of challenging and very small tissues or cells. Gels may take a little more time on the pre-processing side, but they save time on the embedding and post-processing side, and they may also show, give you some nice results for pathology. Described each stage of the procedures for handling demanding tissues from the wet tissue all the way to histopathology analysis. There are many applications for these techniques that we've reviewed today. They're obviously not limited to the ones that we've applied them to, so there's probably a lot more applications you can find in your own laboratories in the cases that you work with. So we advise you to think outside the box, innovate, experiment, and when you're able to, have fun. We have a lot of people to thank. These are the folks from our lab, and they all contributed in one way or in the other to our presentation today and the work that went into it. At the bottom, we have our email addresses, so Jen and I would be happy to answer any and all questions that you might have about the presentation or the diseases that we discussed today, so you are free to contact us anytime. I've also provided some reference mouse pathology texts. Sometimes these have really great images for how to orient tissues that the pathologist might appreciate. And also I've provided a list of references here that we used in this presentation. But anyway, we wanted to thank you for your attention and your time and for attending today.